now. Okay. So now I have the chat box up if you guys have any questions. This might get hard to see. Okay. I think. All right, so we did get into lipids a little bit, right? So we went through this. I talked about fatty acids. So I need to make sure I'm doing everything right on Zoom. I'm screen sharing. I'm unmuted. Awesome. Okay, so lipids. Those are the carbohydrates you were just working on. Like I said, you can make a similar table for lipids if you want to. Remember, lipids don't have distinct monomers. There's not like a monosaccharide equivalent. We talked about fatty acids, and I think this is where I ended, saturated versus unsaturated fatty acids. So again, fatty acids are just these long chains of hydrocarbons, so carbons with hydrogens attached. At one end, you have this carboxyl group, COH, and then a double bond to O. That's your carboxyl functional group. The difference between, you guys told me saturated and unsaturated, is that saturated fats are solid at room temperature, right? So that's like butter. Unsaturated fats are liquid at room temperature. The reason for that is this simple double bond here between a couple of carbons. There can be more than one of these within an unsaturated chain. But if you have all single bonded carbons, like the saturated fat here, that's going to be a saturated fat, and it's solid at room temperature. The reason that this makes just a little bit of a difference is you can tell here by this diagram that double bond makes this chain a little bit bent. So when you have a bunch of these next to each other, a bunch of unsaturated fat molecules next to each other, they kind of separate a little bit because of those bends. If you have a bunch of saturated fat molecules next to each other, they pack really tightly in, and that makes them solid. They pack tightly together, yeah, because they're just one straight chain. An unsaturated fat has this double bond, makes it kink just a little bit, keeps the molecules a bit more separated. Okay. So moving on to a sort of closely related lipid, closely related to fatty acids, because this is basically just 3 fatty acids coming together, is triglycerides. So these are 3, you can see here, these are the fatty acid chains we just talked about. So in a triglyceride, there are 3 fatty acid chains, and they're bonded to what is called a glycerol backbone, which you can see over there in the green. So here we have the glycerol backbone. And that's just a 3-carbon molecule. 1 fatty acid attaches to each carbon, essentially. That's really all you need to know about triglycerides. So if you get fatty acids, if you can identify a fatty acid, you can identify a triglyceride, and vice versa. Triglyceride is just multiple fatty acids coming together on that glycerol backbone. Oh, and it can be a mix of, here you see an unsaturated fat, and then these are saturated. So it can be a mix of saturated and unsaturated in one triglyceride molecule.
I have a question.
Yep.
Okay, did you say there will always be 2 hydrogens connected to 1 carbon in a fatty acid, like as shown on the diagram?
Yep, so those middle carbons, so right here, these middle carbons, there's only 2 hydrogens attached. On the end, you have 3, just because there's a 3rd spot for a hydrogen to attach.
Okay.
Okay, so we've got fatty acids, we've got triglycerides, those are two of our lipids. Phospholipids are ones you're going to be extremely familiar with when we get into cell stuff. So this is our third kind of lipid. It's also similar to triglycerides, except now we only have two fatty acid chains. So we still have this glycerol backbone, which is right here, that 3-carbon backbone like we had in the triglyceride. There's only two fatty acid chains coming off. So for you guys on Zoom, here we have these two fatty acid chains coming off the bottom of this glycerol backbone. And then the really important part of this is the phosphate group. So we haven't seen phosphate in any other lipids yet, but phosphate is another one of those functional groups that I asked you to learn and memorize. That's where you have a phosphorus with 4 oxygens around it. So 4 oxygens around a phosphorus makes it a phosphate, and that's why these are phospholipids. And I'm not really going to talk about the nitrogen group on top. I just kind of ignore that most of the time. So don't worry about the nitrogen group. It's there. The important parts of this really are the phosphate group, the glycerol, and the 2 fatty acid chains. So these phospholipids, because they have the phosphate head and the fatty acid tails, they're actually, so I said lipids overall are hydrophobic, right? They don't mix with water. They're oils and fats. This part, the tails, the fatty acids, are hydrophobic. They don't mix with water. This phosphate head is actually hydrophilic, which means it's water loving. It does mix with water, so the opposite of hydrophobic. So that's going to be really important when it comes to the cell membrane. And we'll get into that, but just know that there are these 2 sides to a phospholipid. It's hydrophobic on the tail side, and it's hydrophilic, or does mix with water, on the phosphate or head side. All right, those are our phospholipids. The last category of lipids is our steroids, or sterols. They go by both names. These are recognizable because you have 4 carbon rings. So organic chemists, when they deal with a lot of carbon, so they don't want to draw a bunch of C's. When they draw molecules, these intersection points, these points on the rings or here, those are all carbons. You don't really need to know that, but in case you're like, what the heck is this? All of these points are carbons. So for you guys on Zoom, if you couldn't see that, this little point's a carbon. Anytime you have a change in direction, basically an intersection of lines, those are all carbons. So carbon, carbon, carbon. Rather than drawing all those C's in, they just connect these lines. And you still have additional kind of carbons coming off the side, you can see. Okay, so 4 rings plus this hydrophobic tail. So this, again, just like the phospholipids, has a hydrophilic side and a hydrophobic side. So one side of it will mix fine with water, the other side will not. And these are important in cell membranes as well, primarily as cholesterol. So I think everyone's probably heard of cholesterol. That's the most common sterol or steroid in our body. And when we get into the cell membrane stuff, I'll show you how cholesterol basically embeds itself in our cell membranes and helps keep them stable. So cholesterol gets a bad rap, but it's really important. If we didn't have it, our cell membranes, the boundary around our cells, would just be all like liquidy and goopy. So we need cholesterol. Testosterone and estrogen, the sex hormones, are 2 other examples of steroids. And that wraps up the lipids. So we've gotten through 2 of the 4. We've gotten through carbohydrates and lipids. We just have 2 left to do in the macromolecule category. I think that's all I have, yeah. So at the end of this lecture, you can go back and look at these just to kind of sort of a review. Like, do you know this information? Can you answer these questions? So these are, it's kind of a mini study guide, I guess, for each lecture. But like I said, I did put together a study guide for the exam. And I think I mentioned this before, but I don't pull information out of the book and put it on the exam. Everything on the exam has been talked about in the lecture, basically. Okay. So any questions about those before we move on, or about the exam, but I'll talk about the details of that more on Friday. Okay. Now, we're going to jump into the last couple of macromolecule types. Oops. There. Proteins and nucleic acids. First, let's do proteins. We're back to having a typical monomer. So everything but lipids has a monomer, that individual subunit, where if a bunch of them are put together, it makes the overall molecule. So for proteins, that monomer is the amino acid. And I imagine you guys have at least heard the term amino acid at some point in your lives. There's actually only 20 amino acids, but we have thousands of different proteins. Any organism, well, not any organism, but higher level organisms typically have thousands of proteins. And they're all made from these same 20 amino acids. The proteins are different just because of the different combinations and sequences of amino acids in that protein. So you mix them up in different ways and it makes a different protein, basically. If we look at one amino acid, just the structure, general structure of an amino acid, it looks something like this. These are just kind of 2 versions of the same thing down here on the bottom. So we have the central carbon. So off of the central carbon, we have 4 things, basically, kind of 4 groups coming off of that central carbon. The amino group, hence the term amino acid. So that's one of those functional groups. You should know the NH2, nitrogen with 2 hydrogens coming off. That's amino, an amino group. There's a hydrogen coming off the top here. That's simple. Off the other side of the carbon, we have one of those other functional groups you need to know, the carboxyl group. That's a C double bonded to an O, and then also bonded to an OH. So carboxyl, all of that together is a carboxyl group. All of that. And then we have this side chain, this R side chain. R is not an element. It just it's a placeholder for any number of different kinds of, different combinations of elements. So imagine this R side chain is just kind of not a random assemblage, but different combinations of elements that make the amino acid what it is. So there's 20 different amino acids. They all have a different R side chain. I'm not going to talk about the details of the R side chain, just know that this is what differs. The R side chain is what differs between each of those 20 amino acids. Otherwise, the amino acids are the same. They have the amino group, the carboxyl group, and that hydrogen coming off of a carbon. Exactly, yeah. So if I show you something that has an R coming off of it, you know it's an amino acid. All right. Continuing on, when we put multiple amino acids together, the bond is called a peptide bond. So with carbohydrates, you were just filling out your table. That was a glycosidic linkage that bonded their monomers together. With proteins, it's a peptide bond. So here you can see one amino acid, another amino acid, you add them together through a peptide bond, and you get what they call a dipeptide. You don't really need to know that, but dipeptide, polypeptide, those are long strings of amino acids. Or dipeptide's only two, but the polymer is called a polypeptide. This is done by dehydration synthesis. So I talked about the reactions being the same. Whenever we're combining two monomers, no matter what the organic macromolecule is that we're talking about, it's always done by dehydration synthesis. So water is always taken out. So here you can see the H2O, two H's and an O, being taken out. And then that's what allows the bond. So that's the dehydration aspect. It dehydrates, takes water out. So here we have the two H's and the O being taken out. That's our water. And then you get the peptide bond. All right, proteins have a lot of functions in our body. Proteins are really, really important, well, in any animal or plant, any organism, basically, any living organism. So I'm going to go through just kind of bullet point list some of these functions. We'll talk a lot more about them, about some of them when we get into getting into the cell information, like the basics of how cells work and how cells are structured. So first off, proteins are really strong. So they're good at providing structure, nice solid structure for different components of living organisms. Keratin, for example, you may have heard of. It's a really tough structural protein that's found in our hair, our skin, our nails. So a lot of like shampoos advertise they have keratin in them to strengthen your hair. I don't know how much that actually helps, but keratin is a real thing. It's a real protein, and that is what gives our hair some strength. Proteins are also really helpful in communication. So you may not think of this, but cells within an organism's body, they're always talking to each other while talking. They're sending signals from one to the other. And proteins, in large part, are what are carrying these signals. So proteins are carrying signals from one cell to another. So a hormone is a chemical signal basically being sent between different parts of a body, between cells. And then, so some of those are proteins, and they also get basically accepted by proteins. So there's receptor proteins on cell membranes that take in that information too. So lots of communication done through proteins. Number 3 here is cell membrane transport. So every cell has this membrane around it, but things need to get in and out of the cell. Usually they can't just move freely. If they could just move freely, our cells would be out of balance. So it's sort of like a tight gatekeeper would be a good analogy for the cell membrane. Proteins embedded in the cell membrane, so something like this, can allow the passage of specific materials in versus out, depending on which way they need to go. So proteins are sort of gates into and out of the cell. We'll talk a lot more about that in the next chapter, I think. And then the last 3 functions, I think these are the last 3 I have listed up here. Proteins, enzymes are proteins. So if you've heard of enzymes, They are molecules that speed up chemical reactions. They don't make a chemical reaction happen if it wouldn't have already. It just makes it a little bit easier. So enzymes help speed up that process. All enzymes are proteins. They're also really important in our immune system. So antibodies are proteins. If you've heard of antibodies, which you probably have over the past year since COVID, we've heard a lot about antibody tests and seeing if you've been exposed to COVID and all that kind of stuff. So antibodies in our system actually recognize and attack any invading cells. So that's what this image is here. These little pink colored, I guess, Y-shaped things are antibodies, and they're attaching to this viral cell, basically attacking it. Say that again. Not be able to fight it? So there's some, like, there are autoimmune issues a lot of times if your antibodies can't necessarily fight something off or they attack the own individual's body. Antibodies are transferred in a lot of different ways from parent to offspring. So if they don't get transferred at some point, you could be lacking in antibodies. But they also, you build them up over time. So when you're exposed to a sickness, you'll then develop antibodies to then fight that virus off, for instance, if you get it again. Yeah. Okay, oh, and then the last one, I forgot about movement. So proteins help in movement. Muscle contraction is a big one. Also in kind of simpler organisms or some of our cells in our body that have cilia or flagella, like those little pieces that move on the outside and help the cell move. Those are all protein-based. Proteins are what's doing that moving. So those are the 6 main protein functions that I want you to be familiar with. Yeah, proteins are probably one of the more important pieces, I would say. I guess all macromolecules are, but proteins have such a variety of functions in an organism's body that they are considered incredibly important from many different aspects. Okay, a little bit about protein structure. I'm not going to go into too much detail about this, but it is important to understand that proteins are sort of complicated in their structure. They start out really simple. So when they're being built, it's just a bunch of amino acids being strung together with those peptide bonds. So you can imagine just a bunch of amino acids in a chain. That's called the primary structure. Just an amino acid sequence. That's the primary structure. A protein is not functional at this stage yet. It has to do a lot of folding on itself, basically, to be eventually functional. So just a string of amino acids cannot do all of those functions I just talked about. The secondary structure, so above here, above the primary structure, it's basically just a lot of folding, lots of protein folding and bonding within that amino acid chain. So the secondary structure, you kind of get this first level of sort of bonding between different parts of this chain, right? So the amino acids are already bonded to each other. Then you get these kind of cross linkages between the chain. So you can get what's called a helix structure. So it kind of starts to wind like that. Tertiary structure, that's our third level. Tertiary structure is basically even more folding. So beyond just a helix, now that helix is folding in on itself some more. Again, just through bonding across the helix. So that's tertiary structure. And then when you get to the highest level of structure, which is quaternary, which means 4, The fourth level of structure, you have multiple amino acid chains that have folded into their tertiary structure already coming together. So you would have, say, this one bonding with a couple other amino acid chains that have already achieved their tertiary structure. So usually, in order to make a fully functional protein, it has to be in its fourth level of structure, its quaternary structure. What's that? Oh, to be functional. For a protein to actually do what it needs to do, it needs to be in its quaternary structure. So if for some reason a protein is built and you have the perfect sequence of amino acids that you need, but it doesn't fold properly, it's not a functional protein. All right. Oh yeah. So just one more slide about the quaternary structure. Oh yeah. So here I put the protein is fully functional at this stage. So this is a stage we need to be at where you have multiple different amino acid chains that have folded over and over on themselves coming together. No, don't worry about the alpha versus beta. They're just slightly different shapes. All right, any questions about that? Any other questions about protein structure, proteins in general? We'll see more proteins in the future. So now you know how they're generally formed. Oh yeah, I wanted to show you this. So this is kind of interesting. So here we have an antibody protein and then a protein from a flu virus. So you can see how the antibody protein, so this would be, say, what you have in your body if you got the flu vaccine. So you have the antibodies for the flu. That vaccine basically tells your body what antibodies to make. So you have the antibody protein, and it matches up shape-wise almost perfectly. It's like puzzle pieces going together. So that antibody can then attack the protein from the flu virus, so it can't replicate anymore in your body and make you sicker. So here is another example where the structure and the shape of proteins is really critical to their functioning. Sorry, I can't hear you. That fan is terrible. Oh. Yeah, so it depends on how much. That's a good question. So for you guys on Zoom, she was asking if the virus evolves. Do the antibodies evolve with it? And yes, ideally, because hopefully the evolution is slow enough that the antibodies can kind of relearn the shape and then continue to actually fight the virus. So with the COVID vaccine, that's what people are worried about with the new variants, that the antibodies in the current vaccines won't keep up with the new variants that are coming out. And they're showing there is, with a couple of the variants, it doesn't seem to be affecting it. And then with, I think, one of the variants, it does decrease the efficacy a little bit of the vaccine, probably because it has gone, that variant of the virus has gone kind of further beyond and changed shape a little bit more. So that's a really good question. But yeah, antibodies, you can think of antibodies as sort of, they're able to learn and change along with new viruses, yeah. Yeah, it'll be interesting to see how that plays out in the long run, for sure. All right, that's good for now. We'll do nucleic acids on Friday and do sort of a little bit of review. I'll use a Kahoot quiz. So if you guys have ever done Kahoot, I like using those as like just an interactive way to sort of review for the exam. So we'll do nucleic acids and then a quick review and hopefully get you ready for the exam Monday.